Hello, inquirers. A quick note before today's podcast for Inquiry. The Centre for Inquiry Canada is a proud sponsor of BajaCon, the Blue Water Atheists, Humanists, and Agnostics Conference, happening in Sarnia, Ontario on August 26th through 28th, 2022. I will be going, and you can too by registering at bluewateraha.com. I hope to see you there. And now, today's PFI episode. Welcome to the Podcast for Inquiry, brought to you by the Centre for Inquiry Canada, a national educational charity supporting your community for reason, compassion, and secular humanist values. You have answers, we have questions. I'm your host, Leslie Rosenblood, board member and secular chair for CFIC. Welcome. On today's episode of Podcast for Inquiry, I speak with Dr. Emma Allen-Vercoe. Emma obtained her Honours Bachelor of Science in Biochemistry from the University of London and her PhD in Molecular Microbiology through an industrial partnership with Public Health England. Emma started studying normal microbes of the human gut at the University of Calgary in 2005. In particular, she was among the few that focused on trying to culture these unculturable microbes in order to better understand their biology. Emma moved her lab to the University of Guelph in late 2007. In 2013, Emma founded New Biota, a research spin-off company that aims to accelerate therapeutic ecosystems as biologic drugs on a commercial scale. Emma and I discuss the importance of microbes to human digestion and overall health, as well as how to think about microbe populations in different people. We talk about the microbiome of the Yanomami, the research that she is conducting with previous Podcast for Inquiry guest David Good, and why this research is important. In addition to hard work, we talk about the role of serendipity in scientific progress and how such an event led to an enormous grant to further colorectal cancer research and the beneficial effects that this might have for many people around the world. We talk about a small improvement that she contributed to on a rather gross but seemingly effective medical intervention, and the seeming ineffectiveness of the probiotics craze, which have transient effects, if any. We also talk about her longstanding campaign against pseudoscience and the hopeful rise of personalized medicine in the years to come. I bring you my conversation with Dr. Emma Allen-Vercoe. Dr. Emma Allen-Vercoe, thank you for coming on to Podcast for Inquiry today.
Thank you for having me.
I've read a few of your works, and I feel like I could talk to you for several hours just to get a basic understanding of the research that you're doing. But I want to start at the very basics. And so I know that you study gut microbes, and I want you to tell me why that's important to people. And to give you a baseline, let me tell you my understanding of digestion. I took biology in high school and a first-year undergraduate course. So my model of digestion is that we take in food, it gets pushed to our stomach where it gets broken down by a lot of acid in the stomach. Once that happens, it gets pushed to our large and small intestines where a bunch of nutrients are extracted, and then the waste products come out as pee and poop. There's more to it than that, but as you've heard, there's really not a lot of room in my understanding of where gut microbes fit in and what function they serve. So I'm hoping that you can fill in some of the gaps and tell me why my understanding is woefully incomplete.
Well, first of all, I'd say your understanding is not that incomplete. I think that actually, if you just have a very basic understanding of how food gets through your gut and makes that journey through, then that's actually a really good start. The missing piece, and just so you know, you're not alone in not really appreciating the role of microbes. The missing piece is the microbes. And the really interesting part of all this is that we really didn't appreciate too much the role that microbes play in digestion until I would say the last 20 to maybe 30 years. I think before that, we knew that microbes played a role. Absolutely, we did. But we didn't really appreciate that the microbes are actually playing a role in our health as well. So, what I mean by that is that we knew that microbes were eating the food that we eat, but we didn't actually really appreciate that as a result of that, microbes were making molecules that our bodies were using to help us with our health. And so that's the really interesting part. But in terms of your understanding of how digestion happens, that's exactly right. You know, you have microbes right the way through your digestive tract. And actually, it's kind of interesting that you have the by far the greatest diversity of microbes at each end. So in your mouth, you have a lot of microbes, and in your distal part of your colon, which is the last part of your large intestine, you have a large amount of microbes. And in between that, you've got variable amounts of microbes, and they're fairly different populations. So your stomach, for example, is a very acidic place. It's not very hospitable to most microbes, and so there aren't that many microbes that live there. But the ones that do are very, very much attuned to their environment, and they're very specialized, and they are able to live in that environment, and they do actually have roles in human health as well. In the small intestine, there's a lot of actual digestion for the human side of us going on. So this is where we process a lot of our food, fats and proteins, and some carbohydrates as well, simpler carbohydrates. And we have very specialist systems in our small intestine to do that. And then everything gets kind of shunted off to the large intestine, all the waste products and all of the undigested food from there. By the time food reaches our colon, which is again our large intestine, or otherwise known as a bowel, it is very, very much different to what it looks like when it goes in. And most of the time, if you're eating a healthy diet, most of the food that makes it to our colon is of the fibrous varieties. This is fiber.
Mhm.
This is the sort of the stuff that we're told to eat more of. Lots of fruits and vegetables, and it's actually fiber. This sort of otherwise indigestible or indigestible to human part of the diet, which is so important to the microbes, because as I mentioned, most of the microbes live in the colon, or a large proportion of them do. You can imagine that if the human body was a planet and the microbes were the things living on the planet, then the colon is kind of like one of the super cities of the body, where we have actually quite a lot of microbial population.
I want to know, what is the difference in microbial populations between people? Like,
Yeah.
perhaps I'm perhaps a little bit taller than you. Perhaps you're a bit taller than me. My hair's a bit curly. Yours is a bit flatter. We are different. Different people have different shoulder widths. Like, what's the right way to think of the microbial population? Because my understanding is that they're not human. Like, they are other separate organisms that live within our bodies.
Yeah.
So, but do we all have the same microbes, and perhaps there's a bit of variation? You have a bit more of type A. I might be missing type B altogether, even though most people have it, but I have a lot of type C, and that kind of makes up for it. Or am I thinking about it the wrong way? Or is something completely unique to you, completely unique to me? Is there differences between men and women, young and old? Are there differences in uh, it geographic populations, like people raised in Japan going to be different than someone raised in Chile, for example? How does it work, and what's the best way to think about microbes, or different populations of microbes in different people?
Yeah, yeah, that is an amazing question and a very, very deep one. And the answer is all of the above. And let me
Okay.
explain to you. So in a very kind of granular way, you can imagine that everybody, every person, even your twin brother or something,
Mm-hmm.
identical twin, would have a different microbiome to you. So your microbiome is very specific to you. And we actually have coined that the poo prints in a way, because when we look at a poop sample of a given individual, we can almost tell, like a fingerprint, who it belongs to, because it's a fairly static thing. So the microbes that colonize and I'm talking about the gut at the moment, although it's probably true as well for other other populations on your body, but the microbes that colonize your colon are very specific to you and are the result of an acquisition that we know very little about. So we're starting to understand that has happened through your life, and for the most part, that's you get populated obviously straight after birth, and then there's about a 3-year development process in normal people, normal healthy individuals, where your microbiome becomes what we call mature. And then that
Hmm.
microbiome more or less stays with you. I mean, there are obviously, it's not completely static like a fingerprint, but from that point of view, there will be changes, and some microbes will come in and some microbes will be lost. And that is very much an effect, or a combination of many effects. One of which is your immune system and the way that that interacts with your microbiome, and that's obviously encoded by your genes. One of which is your diet. That's a very, very big driver. One of which is your geographic placement, and then that obviously plays into the diet that you eat, and also the sorts of microbes that you might come across in your everyday environment. And so there's all of these things are sort of vying for control. So what does this mean? It means everyone has a slightly different microbiome. But the really interesting thing is that although we may have, up to 200 to maybe 500 different bacterial species and many other different microbes in our guts, um, for the most part, the microbes are actually doing very similar things. So even if you've got a very different microbiome to your neighbor next door, um,
Hmm.
you may find that your microbes actually carry out the same kind of work. And that's because of a phenomenon that we call functional redundancy. But what that means is it's just a long word or a long term for basically saying that within our microbial ecosystems, we have multiple microbes that can do the same kind of work. And so together, if you look at the kind of output of an ecosystem that does that, you can't really tell the difference between the work that is done, because if one of those microbes goes extinct in your body, something else that can do the same work can take the role of the thing that's missing. And so outwardly you don't see too much of a difference. So that's very true. And then but when you start to think about geographical things, which is as well driven by diet, you can actually see some big differences between the microbiomes there.
So on that note, the geographical differences, on our last episode, I spoke with David Good, and he mentioned his research, and he was looking into the microbiome, or the microbial population of the Yanomami. But we didn't get into a lot of detail, and I was hoping that maybe you could shed some light on why the microbiome of the Yanomami is of interest. Why bother studying it? What do we hope to learn? And why is it worth looking at this population that has had, comparatively to almost anyone else, really minimal contact with the rest of the world?
Yeah, and that is absolutely fascinating. And as you know, David's now become my PhD student, and so he and I are looking at this, in great depth. And I'm super excited about that, because I don't think that, or I know that without David, I wouldn't be able to do this kind of research. So what we're interested in, and the reason that we're interested in the Yanomami and other hunter-gatherer sort of people across the world, these are people that have lived a very sheltered, if you like, life away from the industrial world. They've lived a very traditional lifestyle, although we don't like to use the word traditional, because it's kind of, it sounds like it's somehow backward. It's not at all. It's extremely advanced, given uh, the amenities that these people have. And the reason that we're interested in it is because we have this hypothesis that was actually coined a few years ago now by Allen-Vercoe and Marty Blaser. It's called the missing microbes hypothesis. And that posits that basically over the years in the industrial world, because of all of the things that we are faced with in the industrial world, and that will be things like the diet that we eat, which is comparatively poor. I think we can all agree the Western diet is not particularly good, as well as the sanitation that we have brought in as a way to reduce the amount of infectious disease, and many other lifestyle events have actually started to erode our microbiome. And so, what we might be seeing, perhaps a little bit belatedly, is an extinction event of microbes in our microbiomes. And the reason we should worry about that is that we actually have very little idea of what these extinct microbes might have done for our health. And what we've the reason that it's kind of alarming is because if you look at the rise of industrialization, and particularly the introduction of antibiotics around the middle of the last century. From that point on, we've seen massive rises in many chronic diseases. And so the hypothesis is that perhaps some of these microbes that we may have lost through things like antibiotic therapy, which of course have been introduced for all the right reasons. We wanted to get rid of infectious disease, but perhaps a side effect of that, the collateral damage, if you like, is that we've also got rid of a lot of microbes that might have been quite important to our general health. And we've only started to realize that recently.
So, how could we tell the difference between uh, the rise? I mean, that we recognize a rise of chronic diseases, I think, is indisputable.
Yes.
But when it comes down to causes, is that because if someone had a chronic disease 200 years ago or more, they simply would have died, or they just would have had some sort of ailment, and everyone had something, so it wasn't recognized as such. So is like how would you be able to disambiguate various possible causes, like whether it would be because of microbes, or that because we are simply recognizing what always was there, but we recognize it now, so the numbers go up, or
Yeah.
people just would have died from them before, and we can treat at least some of the symptoms, if not the underlying causes, so they have a chronic condition, which is bad, but not a fatal condition,
Right.
which would be worse. And like, at least from an outsider's perspective, all of these seem plausible, as well as, possibly we've killed a bunch of microbes that were beneficial, and for whatever reason, the ones that remained, as you mentioned earlier, can't pick up the slack. How can you like go back in time, or do some research now to determine why chronic diseases are going up? And it
Or
could be something
right.
else entirely. I mean, these are just a few hypotheses I'm coming up with off the top of my head.
Yes, yeah, and you're quite right. I mean, the way that we monitor the rise of chronic disease, the incidence of chronic disease, certainly plays into this. And for sure, you can imagine that some of the criteria for measuring disease may have changed over 100 years. Absolutely. However, even what we're seeing seems to be accelerating. And so it does seem to be, in the case of several chronic diseases, that in the last 20, 30, 40 years, we're seeing the rise is just, it's not logarithmic, but it's getting that way. And so we can sort of see that these are that this is problematic. So the problem with this is that of course we can't take everything into account, but we can take quite a lot into account in terms of changes in the way that we diagnose disease and things like that. And so for several diseases, and I'm not an epidemiologist, but I know that they have taken all of these things into account. And despite that, they're still seeing these enormous rises in certain diseases. And so I do believe that it's real, even though
Mhm.
that absolutely there are confounding effects. Statisticians are very good at taking confounding effects into consideration, and even when they do that, we're still seeing these rises in chronic disease.
And so the information that David and you are getting from the Yanomami will help see if indeed there are missing microbes in our
Yes,
guts that
yes.
are present there, and then if we identify them, then we can see, well, what do they do, and are
That's
they
right.
helpful,
That
right?
that's exactly the idea. And so this work is actually some of the most exciting work I've ever been involved in my entire research career. I mean, it's not every day you get to sort of work on these very, very precious samples. What we were trying to do is to understand, okay, well, if the Western world, if the industrial world, is suffered these massive die outs, then that probably started to happen before we had the ability to measure microbes. And so, can we start to sort of go back in time and look at people who have never had some of the exposures to modern life that we've had? And that's essentially what we and others are trying to do. And we've been very lucky to be able to do that with David's family, and we're already seeing very amazing signals of that. So the samples that we've already brought back to Guelph and that we have cultured, we're seeing an amount of diversity in terms of the numbers of microbes that we're able to isolate from these samples, which is far and above anything we've ever seen in even the healthiest, Westerners, Canadians that we've sampled so far. And we're seeing an enormous number of novel species that we've never seen before, as well as some of these what we call missing microbes, which are microbes that have been found across the globe in hunter-gatherer populations, but never in healthy Westerners. And these are kind of the poster child microbes for missing microbes in terms of what do these microbes do, because they obviously are important. They're found in Africa, they're found in South America, they're geographically extremely distant places to each other. And so perhaps these are remnants of our ancestral microbiome that we've lost. And so now we have these microbes in hand, we've cultured some of them. We can start to use our model systems to see, well, what are these microbes doing? Just like you've said, if we understand what they're doing, then we can start to see, well, what are the functions that as a Western population we've lost?
Thank you. Thomas Edison is often quoted. He said something that was fairly common at the time, but it's attributed to him that genius is 1% inspiration and 99% perspiration. But that and I think it's a famous aphorism because there's some truth to it, but it leaves out what often leads to breakthrough insights, and that's serendipity. And can you tell us about how a chance conversation led to a eureka moment for you?
Yeah, so we had this a situation. It was a number of years ago. I was working on a microbe called Fusobacterium nucleatum, which sounds like a Harry Potter spell, but it's actually a very interesting microbe that lives in the mouth. And I was always very interested in it. Nobody had really studied it very deeply because it's actually very hard to grow. It's an anaerobe, and so it's tough to grow, and it smells bad, so nobody really wants to grow it. But we were very interested in it in my lab because we'd read some papers which showed that it actually was, it could be a nasty microbe, it could be a pathogen under some situations. And most people are treating it just like it's one of the microbes that lives in the body, can cause some problems sometimes, probably of no consequence. Well, at the time, I was working at the University of Calgary, and I had some access to biopsy samples from patients with Crohn's disease and ulcerative colitis. And my hypothesis was that Fusobacterium nucleatum may actually be one of the microbes that could be causing a problem in these diseases. And so we actually did that work. It was, like I said, it was some time ago now, but we did that work, and we found that actually, yes, we could actually isolate this microbe from the gut, from these biopsy specimens. And when we did, we found that the nastier strains of these of this species tended to correspond with or the patients who had ulcerative colitis and Crohn's disease, as opposed to our controls at the time. Those were colon cancer screens, so healthy individuals. So that work developed, and I basically, founded my lab on that work. So it's been my bread and butter since. But since that time, I have collaborated. I've struck up collaborations with others, and in particular with a group at the Broad Institute in Massachusetts, in Boston, who were doing who were instrumental in part of the Human Microbiome project. And basically, I was involved in providing them with many different strains from my collection. Fusonucleatum isn't the only thing I work on; many others. And so I would provide them with DNA and they would do the sequencing and we would sort of provide these microbes for the Human Microbiome Project. And then in Canada, I was working on another project that was to do with Fusobacterium nucleatum again, but we had found this microbe, and in fact, this was work that was done at the BC Cancer Agency with Rob Holt, and his group had done some sequencing of RNA. So this is a molecule that's only really present when a microbe is alive, and looking at the RNA signatures of patients with colorectal cancer tumors. And what he had found was that he saw more often than he should do, by chance, the signatures, if you like, for this Fusobacterium nucleatum. So knowing that I worked on Fuso nucleatum, was one of the few people in the world that did, because I was, one of the few people in the world that found it interesting. He contacted me, and we'd sort of struck up a collaboration since, and that work really went places. And so we decided that we would publish this work, which would be a real breakthrough, because basically we were describing a microbe, a bacterial microbe, that could be a causative agent of colorectal cancer, which at the time was a big finding. I still believe it is a big finding. I still think it's very important. So the eureka moment came when I was on the phone with my collaborator from the Broad Institute, and talking about some of the microbes that I had ready for them to sequence, and I was talking about the Fusobacteria, because I was sort of like, well, we have all these new strains of Fusobacterium nucleatum. How many do you want? She said, oh, we should probably get a lot of those, because we've got some really interesting new data coming out of the research that we're doing over here. And this research was happening in the lab of Matthew Meyerson at the Broad Institute. So I said, oh, what's that? And she said, well, I'm not sure I can really tell you, but we've found an association between Fusobacterium nucleatum and colorectal cancer. So I was immediately thinking, oh, wait a minute. We've found the same thing. And I think that when this happens scientifically, usually it's a race to the finish. Who's going to publish first? They take the prize, and the runner-up really doesn't get much of a say so. So I really think the eureka moment for me and for my collaborator, Ashley Earle, who I was talking to on the phone that day, was that we just decided we've got to do something about this, because we don't want that to happen. This is good research. Basically, both labs, so Rob Holt's lab working with me, and Matthew Meyerson's lab working in Harvard, had actually found the same thing. And that actually is a very powerful thing, because it shows that it could be, it's more real. So what we actually
It's independently replicated even before
exactly.
publication.
Exactly. So we were very, very lucky that that Ashley and I knew each other. So we hatched a plan, basically, to go back to our respective collaborators. So me to Rob Holt, and Ashley to Matthew Meyerson, and say, hey, for Ashley, it's like the Canadians have got the same result, and I went back to Rob and say the Americans have got the same result. And to their credit, both Matthew and Rob got on the phone with each other, and decided that the best way to deal with this was to publish the results back to back. And that made it a much more powerful story, because, in fact, there were elements of both stories that made the whole story stronger. And so we did, and we published back to back in Genome Research, which is a fairly large journal, some years ago now. And the result of that has been amazing, and a really good. It's been great for me from the point of view of it's bolstered my faith in research and how it should be done. It should never be competitive. It should always be collaborative. And I say that because some really great things have come out of this relationship. First of all, this became a big story, and now all of a sudden everyone studies Fusobacterium nucleatum, which is great for me, because I've got probably the largest library of fusonuclear strains that exists in the world, and so I collaborate with a lot of people now, and everyone is finding the same thing. So this seems to be a universal thing across countries, and it's been really gratifying to see that this isn't just sort of a flash in the pan hypothesis, that this is real. And the second thing that happened that's so exciting is that a number of years later, Matthew Meyerson led a consortium of really high-powered researchers to come together to write a grant for one of the Cancer Research UK's grand challenges awards, to the really, really big grants, multinational, multi ideas. So him and Wendy Garrett, so Matthew and Wendy Garrett, who's also at Harvard, co-led this application. And Wendy's been, far away one of my top 10 collaborators throughout my research career. She's an amazing, and so all of it, and I'm one of the applicants, and as you can probably guess by the smile on my face, we got the funding. So this is an enormous help. This is an incredible funding opportunity where we can come together as a group of, I think there's 13 principal investigators, of which I'm one. They cross from so we're in Canada, we're in the US, we're in Spain, we're in the UK, and the Netherlands. And all of a sudden, that has not only given my lab, a much needed injection of research money, which is always a great thing, but my trainees have been able to actually go to international labs and learn with the best people how to do some of the best science, and it's such a great story from that
So,
point.
what are you trying to learn from this research and from this grant? Like, so you mentioned that there's this association, and because of the association is strong and independently verified, that it's likely a causative factor of this particular microbe and colorectal cancer. So that's good.
Mhm.
Are you looking at early diagnosis? Are you looking for more accurate screening for people at higher risk? Are you looking for ways to treat the cancer so that if people get colorectal cancer, they'll be able to be better treated with better results and fewer side effects?
Yes.
What are you trying to achieve
Yeah.
with this wonderful grant and this international collaboration?
Yeah, so I think that this is a great lesson again for me on how amazing science can be when you're given a large sum of money and you put a group of experts together in a room who are not all experts on the same thing, but experts across the board. So the grant is called Optimistic, and off the top of my head, I can't remember what that stands for now. Is Optimistic with two C's at the end, but it's opportunity to investigate the microbiome in colorectal cancer. It's that or some semblance of those words arranged into a nice sounding acronym. So the answer to your question is all of the above. This is one of the few opportunities when you have these enormous grants like this where you can look at the problem, and the problem is colorectal cancer and microbes or the microbiome in colorectal cancer. And so this is one of those opportunities where you can look at that problem through multiple different lenses within the same group, and that's huge. We get together, once a month as a group online. There's 130 people at these calls, and there are always 130 people on these calls. There's one of them better, even through the pandemic, one of the better, attended Zoom calls I've ever been on. But they're always fascinating. So what we do is split the work up into 6 what we call work packages, and each one of those work packages is led by a different consortium of these 13 researchers, although some of us work on more than one. And we work on different aspects of it. So we are looking, for example, for biomarkers, and so the team with bioinformatics expertise and with sample collection and clinical trials expertise are taking samples and trying to understand whether or not we can see a signature of what we call oncomicrobes. So Fusobacterium nucleatum started it all, but honestly, there are now several microbes that we recognize that have a role or seem to have a role somehow in the development or the proliferation of colorectal cancer. And we've dubbed them oncomicrobes, cancer-causing microbes. And so we're looking at all of those now. So we've broadened the focus somewhat, which I think makes it more powerful. So we're looking for these signatures, and actually in the UK consortium there have been looking at the fit tests, or the tests that we sometimes are asked to do, or we should all be doing, where we put a bit of poop on a card and send it back in the mail or whatever. So we can actually look at those for microbial signatures, and when we crowdsource that, when we have a lot of data, then we can start to see signatures of colorectal cancer. And so that's what they're doing. On the other hand, there are other groups of us who are trying to develop model systems, and that's what I'm involved in. So, can we actually create model systems in the lab, be that in a mouse or in a bioreactor like we do, or whatever, to prove that these microbes are causative of cancer? Because when we have a model system, then we can actually start to intervene and see what's going on. And then in the last work package that I'm involved in, we're actually doing some cool stuff where we're trying to see if what are the next generation therapeutics that we can develop. So, for example, if Fusobacterium nucleatum is an oncomicrobe, can we vaccinate people against carriage of the particularly nasty strains so that they don't develop colorectal cancer in the future?
So like the HPV vaccine,
Yes,
you
exactly.
might be able to vaccinate people against cancer, because that's something that it's very rare. Most vaccines are against uh,
Infectious disease. Yes,
bacteria or viruses,
that's right.
but
Yep.
to be able to vaccinate folks against cancer, that would be amazing. And then we'll be able to identify, if you're successful on all fronts, who's at the greatest risk, so that
Exactly.
if we can't, or for whatever reason we shouldn't vaccinate everybody, we know who the right target population is.
That's exactly right. And that's where a large consortium of researchers like this, with a large injection of money, can make enormous impact, far more impact than you can make with, a little bit of grant money. And then you sort of publish a paper, and then you just take the risk that someone's going to read that paper in the right place at the right time, and science goes on. Here, we're actually in this cauldron of really great research where whatever we do is linked intricately to whatever everyone else is doing, and we're all aware of what everyone else is doing. And I think that that's the real difference. And so we're starting to see some real results coming from this, and there's and we're having a lot of fun doing it as well, because, as I said, having these research collaborations really enriches. Certainly enriched my research career. So there's me in this little in this little corner of Canada now, interacting with some of these really great minds across the world, and I think that's the only way that we can really make an impact in these a really solid impact in these kinds of big diseases.
Prior to speaking with you, I thought I had never heard of you, but that's not entirely true. I knew indirectly about one of your research papers, because I'd heard about it, uh, and it was about a new medical technique that seemed to work, and so that's why it was in the news. But it just had an enormous yuck factor for me, and apparently for everyone else, or at least a lot of people, and that was taking the poop, the stool from a healthy person, and then sort of injecting it into the digestive tract, into the colon of someone who was sick, and it seemed to be effective. But your paper, which has a terrible pun that I absolutely love, called repopulating the gut, you demonstrated a proof of concept for something that was similar, but it had significant differences that removed at least a big part of the yuck factor. Can you share some of the details of your research, and what the effects have been?
Absolutely, yes. So what you're talking about is something that we actually call fecal microbial transfer, or fecal transplant, FMT for short. So we don't have to use the fecal word, because then patients are afraid of that, I think. You're right. It is kind of disgusting. And I got into this space again through serendipity, through a colleague of mine called Elaine Petrov, who at Queen's University, who is an infectious diseases doctor, who had been working using fecal transplants. She was one of the few doctors in Canada at the time who tried these out. And her story was awesome. I'll briefly tell you in a nutshell. What happened was, she thought that fecal transplant was, pretty gross. But she once had a patient that started it all off, who essentially begged for it. And this patient had a very serious infection, a Clostridioides difficile infection, which had really progressed to become something that we call toxic megacolon, which sounds like a superhero, but actually is a really sort of end stage disease for this kind of thing. And this patient was dying. And the patient had obviously heard about fecal transplant and wanted to give it a try. Now, as an infectious diseases doctor, the idea, Elaine says, of actually giving a patient who was that sick, feces from another individual, feces we think of as dirty, it's full of germs, full of nasty things. She thought it's the last thing that she should do,
Right.
but because
You don't want someone else's waste products.
that's right,
It's waste,
exactly.
right? Your body ejected it for a reason.
That's right. Using it as medicine just seems like such a foreign concept, right? And so I think she did it under duress because apparently the family and the patient were, basically begged her to do it. And the upshot of it was, to cut a long story short, is that patient recovered and became the poster child for health in terms of gastrointestinal health. And she couldn't believe it. Honestly, she would sort of see him from time to time and see he's doing really well. And so she started to sort of rethink her ideas about fecal transplant, and perhaps maybe this should be a sort of examined and or at least sort of looked at in a little bit more detail, that it might have good potential. And there are other doctors at the same time across Canada and across the world who are seeing the same thing. So this as I said, it's not a new technique. In veterinarians have been using this for a long time to kind of jumpstart the guts, particularly ruminant species that get sick, because obviously the ruminant gut relies on microbes far more. And I think if you look back in history, I think there's a sort of famous period in history. I couldn't tell you how far back, but quite some time ago in Chinese medicine, talking about using yellow soup to treat patients with gastrointestinal diseases. And that yellow soup is as you can imagine, some kind of feces. And so not particularly appetizing, I'm sure, but it seemed to do some good work. So Elaine, after she'd done a few fecal transplants, and she was working with a lot of Clostridioides or C. difficile patients. And C. difficile is a nasty, nasty infection that you can get if you are exposed to antibiotics. What seems to happen is that those antibiotics that are used to treat whatever else it does, it's usually it can be a dental infection, anything like that, but they have a knock-on effect on the gut, and they can start to kill off the microbes that live in that important part of your gut, the colon. And when they do so, if you happen to also be colonized by this C. difficile microbe, or you happen to pick it up from being in the hospital, it can kind of proliferate and take over. And when it does that, it causes some really nasty stuff. So fecal transplant is a way of putting back some of those microbes that you might have lost, so that they can then sort of control the C. difficile organism. But obviously, it's poop, right? And as we said, poop is a horrible thing to imagine as medicine.
So
So
what was your improvement on
yeah.
poop?
Yeah, so Elaine actually was the one, and she came to me. She was aware of the fact that I was getting quite a reputation in my lab was getting quite a reputation as able. We used to be able to say we could culture anything from the human gut, which is um, actually a really important thing to get across. I've kind of mentioned it, but I should mention it again. Most of the microbes that live in the human colon in particular are very, very tricky to grow, because they require very specific conditions. For the most part, they're anaerobic, which means you can't grow them in the presence of any oxygen at all, and so you need very specific, specialized equipment. Well, we have that equipment, and we'd kind of created these libraries of strains that we've been starting to collect, since my days in Calgary. And so Elaine knew this, and so she asked me at a meeting once. She said, "Do you think that I could get some of your microbes from your library, so that we can just give it to patients as pure microbes, so we don't have to use poop anymore?" So at the time, I was thinking, well, okay, yeah, not a problem. But I actually thought that the better way to do it was to find a healthy donor, and to do the culture work from that one donor, so that those microbes that we isolated will have seen each other before. They'll know how to work together, and that's
So
really
rather than taking something that's over here and something
yeah,
that's over here and something that's over here and something that's over here, and then
yeah,
combining them and injecting, you're taking it where a healthy collection were already
yes,
together,
that's
and
right,
so they move essentially as a group,
that's right,
although
exactly right.
purified, so that it's not just.
That's exactly right. Yeah, we feel like nature has done the hard work for us. So why should we, think in our arrogance that we know which microbes should work best together? We should just take a healthy donor and say, okay, they're healthy because they've got the right microbes. Well, let's try and isolate some of those microbes, and that's what we did. So we isolated quite a lot of microbes from this individual, the donor that we found. And in the end, we picked, I think, 33 strains to put together. And we grew those all we grew them. separate microbes so that we could check to make sure that that's all they were, and that they weren't contaminated with anything else. And this is the real magic, was always that in fecal transplants, you can test for what you know, but you can't test for what you don't know. So you have no idea half the time about all those things that you're putting in there, what's in there, and that's where the danger of the procedure comes from. So by cleaning up, basically we're giving a cleaned up fecal transplant, and much reduced one, because we're providing a lot fewer microbes than perhaps you would get in stool. But we thought, well, we'll give it a try. And so Elaine organized to find 2 patients who had C. difficile infection, very serious C. difficile infection that had become recurrent, meaning that they were constantly on cycles of antibiotics to get rid of the C. diff. And then as soon as they stopped the antibiotic, the C. diff would come back again and cause, more problems. So these patients really had no other option at this point, other than stay on antibiotics. And this is a nasty infection, as I said. It can be very debilitating, and it usually affects elderly people more often than others. So these patients were really not in the best of state. So in my lab here in Guelph, we grew up these microbes. We purified them. We checked to make sure that they were pure. We did everything we could as aseptically as possible. We put them together in sort of like a soup, if you like, of just a buffer. So it didn't look like stool at all by the time you finished. It just looked like a very weak milkshake, white milkshake. And we packaged that up into syringes, and we in anaerobic pouches to keep everything anaerobic, and then we sent them via a courier, and the courier happened to be my husband. And the reason for that was because we could have sent them with a courier, like a courier company, but we figured that this was a lot of work that went into this, and we wanted to make sure that this package didn't get lost or thrown or anything like that. So we basically packed it in the back of my husband's car, and he drove it all the way to Kingston, and it went into the patient the next morning.
So it
And
was like an injection, or
it was given via enema. So,
okay.
sorry, via colonoscopy, so a little bit further up the gut. So, but the idea was always to give it by enema, and we started off via colonoscopy because we figured that the further up we could get, we could kind of like spray it through the whole gut and really kind of seed the gut.
Mhm.
And so Elaine did that, and it worked for both patients. Both their infections went away completely. Their quality of life went up immeasurably. And although they were both fairly elderly, and they really did improve enormously in their health. And so we thought to ourselves, wow, that's great. So that's when we decided that we would publish a paper. And the name, by the way, I should credit Gregor Reid. He's a probiotics researcher at the University of Western Ontario. And I was telling him over dinner one night about what we were doing, and he said, "Oh, you should call it Repopulate," and the name stuck. So that's how these things happen. I'm not clever enough to come up with something like that. I need someone like Gregor.
You mentioned really off the top that our microbiomes are stable throughout
Yeah.
our year. Like they from birth to 3 years it grows, but then it's relatively stable throughout. So I'm assuming, based on our conversation, that that's absent a major external event. So if you take a huge dose of antibiotics,
Yes.
which might be for a very good reason, but you take a huge dose of antibiotics, that's going to change your microbiome, and when you get off the antibiotics, it might not recover in the same way that it did from before. Or when you deliberately seed someone else's microbiome or certain strains of things, that do these have temporary effects, or do these have lasting effects when you have these significant external interventions
Yeah.
on what would otherwise be a relatively stable population?
Right, right. That's the $1 million question right now. It's fascinating to me because so the idea here, going back to what we were doing with our patients, is that we would colonize our patients with these microbes, and those microbes would kind of hang out there forevermore. Um, Now that more and more people have done these kinds of fecal transplants, and studies like ours, what we're seeing is not quite that. It's almost like there is a pressure for some of those microbes to be lost. So they act, if you like, as a sort of like a band-aid. They help the patient to get over whatever it is that's causing the problem, in this case, C. difficile infection. But then they don't seem, or some of them anyway, seem to be lost completely. And we don't really know why that is, but it's probably a combination of many things. And one of the things that we're very interested in my lab is, although we're not really studying this as much as I'd like to, and I think other people are, but we think it's to do with the way that the immune system is set up. So that early stage I mentioned in life, when you start to become colonized by microbes and you're picking them up from your environment, the microbiome is fairly transient. It's changing very rapidly during that time. And then around the time of, like I said, about 3 years old, it becomes stable. That coincides with the time that the immune system is sort of becoming more and more mature and stable. And so I don't think that's a coincidence. I think it might be that the microbiome is becoming in tune to your own immune system and vice versa. And so that bond, if you like, is quite important. And now when you're sort of adding microbes into the mix with a fecal transplant, perhaps the immune system is seeing some of those things as foreign visitors and getting rid of them, only tolerating them if and allowing them to stay and to colonize if they're really providing an important function that the body couldn't do without, and then sort of like adapting to allow that microbiome, that microbe to proliferate and live within the body. And we know so little about that. It's extremely difficult to study. Obviously, long term, we're talking about many months and years and down the line, and so it's very hard to do those kinds of studies. But we already know, for example, from probiotics. A lot of people are familiar with probiotics. These are microbes, live microbes that you can
Mm-hmm.
take. You can get them from the pharmacy or from the health food store, and you can take them. And the idea is that these are going to colonize your gut and give you beneficial effects. Well, actually, they don't colonize, and we've known that now for some time. That whatever effects they have seem to be transient as they're passing through, if they have any effects at all. And we could have a whole conversation about how many probiotics are actually not really worth anything at all. There are a few which have been clinically proven, but because of the way that probiotics are regulated, it makes it very difficult to tell as a sort of an educated layperson which is the right probiotic to take. And so we know that these microbes are just passing through, don't necessarily colonize, and that makes sense because, as I mentioned, we've got a poo print. So if everything that we ever met sort of colonized us, we'd have a very different microbiome going through life. And so trying to understand what drives that, how do we pick the friends that we spend the rest of our lives with in terms of our microbes, and how do we tell if something isn't useful, and how do we get rid of it? We don't know. And I think that's a really, really fascinating part of the research portfolio right now.
Okay, I want to be respectful of your time, so rather than ask the 20 questions that I have for you, I've got just I'm going to put the others aside and just ask one more, but it's not necessarily a quick one. I want to zoom out a little bit, and I'd like you to speak not about your research in particular,
Okay.
and I want you to speak uh, to the interested layperson, to people like me, to people like the listeners of this podcast. And so every few years, I hear about this revolutionary new way to think about diet, nutrition, or health, and it's all very scientifically valid and proven and rigorous according to the media reports, which is how I get information. And whether it's a low fat diet from like 30 years ago, or a low carb diet from about 20 years ago, or whether it's macronutrients or micronutrients or probiotics, which was a really big thing like 5 or 7 years ago. And now I'm hearing about microbiomes that might revolutionize how we think about health. I'd like your help and your insight on how folks like myself and our listeners can stay informed and avoid the hype and recognize the charlatans who will use all the same terminology that you do but don't have like the actual research, and what they're hyping don't have the beneficial effects that they're claiming.
Yeah, that's a huge thing, and I have been a big proponent against pseudoscience for a long time. I am very concerned by the amount of pseudoscience that we have in society right now, and how much it informs people's decision making about what their nutrition, etc., etc. Now, I'm not a nutritionist, but I would say that we know, and I think most people can conceptualize this, that the Western diet is pretty poor. One of the reasons why we consider it and I'm talking about the average Western diet, that it's poor is because it's very fiber poor. So we're understanding more and more that fiber is very important for gut microbiome health. However, the problem I think in the popular press and with pseudoscience, with all of this, is that everything becomes very prescriptive in terms of everything is very generalized. So all of a sudden you'll hear about, hey, we should try the Atkins diet or whatever, right? And then everyone tries the Atkins diet. And the thing is, what we have to remember is that everyone has a different microbiome. And so what is right for you is not necessarily right for your next door neighbor or your brother or whoever. So we have to start to understand that. So I should mention this as well, that we have a genome. We know we have a human genome, but we also have a human microbiome genome, and that we call it our second genome because it actually codes for about 100 times more genes than our actual genome does. And it's going to be subtly different between everyone because everyone has a different microbiome. And so it's like everyone is attuned to a different diet, a different set of conditions, and we need to optimize that to the best of our ability. So unfortunately, medicine at the moment, as well as pseudoscience and nutrition, everything is not set up in an individualized, personalized way. And you ask the question, well, how do you know what's pseudoscience, or how do you know what's real and what's not? I think that every time you hear about the new, well, we shouldn't eat this because it causes cancer, or we shouldn't do this because we should drink red wine because of this, you have to remember that that might be based on like one or two studies that is maybe done on a few hundred people at best. And that doesn't mean that it's true for every single person because everyone has a different microbiome. When we do this research, it's a kind of a joke in the field that we're in. That every time we do a microbiome study, we encouraged to report our results, and we do. We report those in scientific journals. But if it gets picked up by the popular press, oftentimes they want a quick, something that sells, and so they'll have this sort of uh, bite of information, something like, if you eat bacon, it's going to give you cancer, this kind of thing, which is totally not true, and actually distilled down to the point where it's almost nonsense. Where there might be a kernel of truth in it, it doesn't mean that it can be applied to everyone. So we have to be very careful when we read these kinds of studies. And so if you see that, and you see, okay, well, if you see a report that says that, if let's say if you drink green tea, then that's really healthy, then you have to remember that, okay, the research that did that from where that originated may have shown that there were some health benefits from polyphenols from green tea. And it may have shown that in mice, or it may have shown that in people, but a smaller number of people, or whatever. It doesn't mean that it's true for everyone because everyone has a different microbiome. So I think that the answer is you just have to take everything with a little grain of salt. And also just try to remember that your microbiome has evolved to protect you in a way. It's a second genome. So it's your job, if you like, as the host of your microbiome, to provide the right environment for your microbiome. And so what do I say, you are your microbe manager. You're like a zookeeper for your microbes. And so the best thing that you can do, if you were in a zoo, an actual zoo, and you were a zookeeper, you wouldn't start feeding all the animals on food that they shouldn't really eat, and additives and things like that. You would provide them with the best possible diet, right? Known from the science that and so you have to sort of start thinking about that from your own point of view. The microbes that you have require lots of different substrates. So you should eat a diet which is healthy, full of fiber. We know fiber is good, but there are other things as well. Good proteins. We need variety in our diet as well. So we shouldn't all just be eating the same thing day in, day out. And of course you should listen to your body. So if you find that you can't tolerate wheat or something like that, then don't eat it, then that's fine. But we shouldn't just copy. There was this great thing about this sort of wheat belly thing a while ago, so that everyone stopped eating wheat, and I don't think for a minute that everyone couldn't tolerate wheat. It's just became a popular fad. And so I think we
Right.
have to listen to our bodies, for one. And yeah.
Could something like wheat intolerance or lactose intolerance possibly be treated by repopulating the gut with appropriate microbes, or is that a way? Is that overstretching what is even a reasonable extrapolation from the type
Yeah.
of work that you're doing?
Well, absolutely. I think that so lactose intolerance for sure. We can think about in the future having microbes which can process lactose and provide them in part of the diet. But we actually have lactase and things like that, so we know the enzyme for that, so we can do that much more easily. In terms of wheat intolerance, it's a bit tricky because then we have this situation of celiac disease, which is a very serious condition and is caused by a total intolerance to wheat, to the gluten protein. And then we have this sort of like fairly ill-defined, wheat intolerance. Well, what does that actually mean? Does it just mean that it makes you feel a bit kind of bloated every now and again? That's not the same as celiac disease. It is totally possible that wheat intolerance that is perceived maybe because you might be missing a microbe that can process a type of glucan in the wheat or something that would prevent that glucan from being an irritant to your gut. But it's going to be different for every single person, and it's very, very difficult to kind of measure and study. And so, it's almost easier to say, well, okay, if you find you can't eat wheat, then don't eat wheat. There's plenty of other things that you can eat. So I think that's the answer there.
Right. It reminds me what you said brought to mind. Ben Goldacre is a British MD and science communicator, and he titled one of his books, "I think you'll find it's a bit more complicated than that." And
Yes.
I think it's a pretty good maxim. It seems similar, consistent with what you were saying.
Yes.
I want to thank you, Emma Allen-Vercoe, for your time. Any last thoughts for our audience today?
I would just say, as I was mentioning, I think it's very, very important these days for us to be far more in tune with our bodies than we are. I think the problem is that we'd much rather listen to what it says in a magazine about what we should be eating than listen to our own bodies. And I think perhaps we should all be a bit more mindful about what we're eating and about how it makes us feel. And I think if we were to do that, then I think that would solve a lot of the problems that we have that we have right now.
Well, thank you for taking the time to speak with me today and to
You're very
speak with
welcome.
our audience. And thank you for the research that you're doing that will benefit us all. This has been a great pleasure. Thanks very much.
Thank you.
Bye bye.
Bye bye.
We hope you enjoyed today's episode. Links for today's discussion can be found in the show notes. If you enjoyed today's podcast, please subscribe and engage in the conversation. Comment, rate, and review. Email us at podcast@centerforinquiry.ca. We'd love to hear your perspective. Podcast for Inquiry is brought to you by the Centre for Inquiry Canada. We rely entirely on donations to be your voice supporting science, free inquiry, critical thinking, and secularism here in Canada. To our supporters, thank you. If you have not yet contributed, please consider making a donation today at centerforinquiry.ca/donate or becoming a member for only $30 per year at centerforinquiry.ca/join. Your contribution supports our efforts to have reason and evidence drive decision-making everywhere. CFIC is on the web at centerforinquiry.ca. We are on Facebook and follow us on Twitter at CFI Canada. Podcast for Inquiry is produced and edited by Matt Payne, Nikolay Nikitushkin and Martin Zelinsky. Music by Anthony Lazzaro. I'm your host, Leslie Rosenblood. See you next time.